What researchers just found could quietly change how we think about MS, and almost nobody is talking about it. And no, it's not a miracle cure, and it's not doom either. If you're new here, this channel is where we break down MS simply, with no doom, just truth. And if you're living with MS or trying to understand it, you're in the right place. This channel exists to make MS make sense, with no doom, just truth. Because MS is confusing enough, we don't need the internet making it worse. And what they just found, it's one of those, wait, that changes things moments. Tap subscribe so you don't miss the full breakdown, starting now. Okay, so here's the weird part. In our world of constant information, we're used to science news hitting us like a fire hose. Every day there's a new study about coffee or red wine or a miracle vegetable that promises to change everything. But the research we're about to unpack didn't make those big, splashy headlines. There were no dramatic news alerts pinging on your phone, no primetime specials. You definitely didn't see any breaking MS cured by broccoli articles flooding your social media feed. And that's precisely what makes this so interesting. Because sometimes the most profound discoveries aren't the loudest. They're the ones that quietly rewrite our entire understanding of a problem. And yet, this might actually matter more than all of those combined. Here's what a team of researchers found. They looked at the immune cells that are supposed to keep the Epstein-Barr virus in check. These elite soldiers called CD8+ killer T cells. Their job is to find and destroy virus-infected cells, acting like a highly specific security force. But they found these cells act very differently in people with MS compared to people without it. It's like comparing a disciplined patrol to a frantic, over-caffeinated squad that can't tell friend from foe. Digging deeper, they notice one specific Epstein-Barr virus gene essentially flips on in the immune cells of people with MS, and crucially, it stays quiet in healthy individuals. Think of it like a light switch for the immune system that gets stuck in the on position. This keeps the system perpetually amped up, constantly on high alert, which is a key ingredient for autoimmune trouble. And this led to the biggest piece of the puzzle. They discovered that MS may be heavily influenced by something called cross-reactive T cells. These are the immune cells originally trained to hunt for EBV, but due to a case of mistaken identity, they also recognize and attack targets in the brain and spinal cord, specifically a protein in the myelin sheath that protects our nerves. It's a biological case of friendly fire on a microscopic scale. To put it in everyday terms, this is the scientific version of finding out your phone battery drains so fast because you have 47 apps running in the background that you forgot you even opened. It's not that the battery is necessarily broken, it's that it's being exhausted by a hidden, persistent process. And for the first time, scientists have a clear picture of exactly which apps are causing the drain. This goes against what we thought was happening, what we thought immune attack only, damage only, one direction decline. What this suggests, something reversible, something modifiable, something earlier. This is like thinking your car engine exploded. And then someone says, actually, your parking brake was on. Here's the part that made me stop scrolling. T cells trained on EBV can mistake a brain protein, anoctamin-2, and attack it. That's a direct immune misfire. Plus, an EBV gene flips on mostly in MS, which may be goosing those overeager T cells. If this is true, it means MS might not be just random immune chaos. It could be a misaimed response with a clear trigger, and that is a big deal. That's like finding out your GPS wasn't broken. You were just in airplane mode for 6 years. You know me. We don't do hype here. This does not mean cure tomorrow, stop meds, miracle therapy, everything reversible. But it does mean researchers are looking at MS differently. This isn't we solved MS. This is more like, hey, we found another piece of the IKEA instructions. So, we've journeyed through the science, connecting the dots between the Epstein-Barr virus and multiple sclerosis. But this isn't just an academic exercise. If you or someone you love is living with MS, you're probably asking the most important question. So what? What does this actually mean for me? Let's break down why this groundbreaking research could fundamentally change the landscape of MS care. First, it reframes the entire concept of timing. We've long suspected that MS has a silent phase, where the immune system is already mistakenly attacking the brain and spinal cord, but symptoms haven't become obvious yet. This research suggests that misaimed immunity, potentially kicked off by EBV years or even decades earlier, is the slow-burning fire that eventually leads to the symptomatic blaze. If we can identify people at high risk during this silent phase, perhaps through specific EBV antibody markers, it opens the door to preventative strategies. The idea of treating MS the first relapse before significant damage occurs moves from a distant dream to a tangible goal. Second, this gives brain repair research a massive boost. For a long time, the focus was on stopping the immune attack. But what about fixing the damage that's already done? Understanding that EBV-infected B cells might be driving the damage gives researchers a more specific target. It helps them ask better questions. If we can remove these specific cells, does the brain's natural repair process, called remyelination, kick into a higher gear? This shifts the conversation from just managing decline to actively pursuing recovery, because we're learning that not all damage is a one-way street. Third, it validates a new class of treatments. Ideas for a targeted immune reset are moving from theory to real clinical wins. Specifically, we're talking about therapies that target Bruton's tyrosine kinase, or BTK. Think of BTK as a key ignition switch inside those rogue B cells. By turning off that switch with a BTK inhibitor, you can calm the cells down without wiping them out completely. This is a more nuanced approach than some older, broader immune suppressants. And it's working. In a landmark trial, one oral BTK inhibitor became the first therapy in over a decade to show it could significantly reduce disability progression in primary progressive MS when compared to the existing benchmark. For a form of MS with notoriously few treatment options, this is a monumental step forward, directly born from this deeper understanding of B-cell biology. Fourth, we're understanding progression better. This research helps explain the frustrating variability of MS. It's not one size fits all, and it's not one speed fits all. The disease isn't just about sudden relapses. There's also a smoldering inflammation that can drive steady progression. The EBV link suggests this smoldering fire might be kept alive by those infected B cells hiding out in the central nervous system. This helps explain why some people improve, why some plateau, and why others experience a slow, steady worsening of symptoms even without clear relapses. MS is basically the choose-your-own-adventure book, except you didn't choose it, and all the pages lead to a place you'd rather not be. Understanding the author of the story, in this case EBV's role, gives us a chance to finally rewrite the ending. Finally, this has huge potential for diagnostics. Getting an MS diagnosis can be a long, anxiety-filled process of ruling other things out. There are several look-alike conditions like neuromyelitis optica or neurosarcoidosis that can mimic MS symptoms and even MRI findings. What's promising is that specific EBV antibody patterns appear to be highly unique to MS. This could lead to a blood test that doesn't just suggest MS, but helps confirm it with much higher certainty, separating it from its mimics and providing clarity when the clinical picture is murky. As always, it's crucial to remember that none of this information replaces the guidance of your neurologist. Instead, think of it this way. For years, doctors have been fighting MS with a limited toolkit. This research doesn't just add one new tool, it provides a whole new blueprint that will help us build better, smarter, and more effective tools for years to come. This isn't just one study, this is part of a bigger shift. Researchers are moving toward repair, protection, prevention, immune reset, brain resilience, less about stopping damage, more about helping the brain recover. Old approach, stop the fire. New approach, stop the fire, rebuild the house, and install sprinklers. This is the weirdest part. This kind of research doesn't get clicks because not dramatic, not cure, not scary, not hype, but it's the stuff that actually moves things forward. Headlines prefer cure found. Science prefers we observed a statistically relevant cellular interaction. So what happens now? More research, more trials, more targeted therapies, more understanding of progression. This is how breakthroughs actually start. They don't start loud, they start quietly, like this. Science breakthroughs are like MS symptoms. They show up subtly, and then you're like, wait, that's new. If you're living with MS or you love someone who is, you know the information fire hose can feel overwhelming. There's so much noise, so much fear, and often so little clarity. That right there is the entire reason I make these videos. It's not just about reporting on the latest study. It's about translating the science into something that makes sense, something that empowers you instead of frightening you. It's about finding the signal in the noise. Because real, meaningful progress is happening every single day in labs around the world. These breakthroughs don't always make the primetime news. They don't trend on social media. They're quiet, methodical, and built on decades of dedicated work. But this quiet progress is what chips away at the foundation of this disease. It's what turns a terrifying mystery into a solvable puzzle. And the more we understand the how and the why of multiple sclerosis, the less power it has over us. Knowledge replaces fear. When you understand the mechanisms, like the role of EBV or the different progression pathways we talked about, the unknown shrinks. It becomes a condition you can manage, a challenge you can face, not just a shadow you have to live under. Let's be honest, MS is still incredibly confusing, but it's getting less mysterious with every single publication. Each study is like a pixel in a massive image. By itself, it might not show much, but when you put them all together, as we've tried to do today, the picture becomes clearer. We're moving from a place of pure mystery to a place of complex but understandable biology. So after all that, what are the huge takeaways? What did researchers just uncover that truly changes the game? First, they've solidified the link between EBV and the immune system's misfire in MS, giving drug developers a much clearer target to aim for. Second, they found evidence that MS might not be one disease, but at least two distinct paths right from the start, which could revolutionize diagnosis and treatment personalization. And third, they're confirming that we can and should target the slow, smoldering progression, the PIRA we discussed separately from relapses, opening the door to a new generation of therapies. If this deep dive helped you connect the dots and made the world of MS research feel a little more accessible, please take a second to hit that like button. It's a small click for you, but it tells the algorithm that this is valuable information, helping other people who are searching for answers find this video. It genuinely makes a huge difference. And if you want to keep up with this journey to have multiple sclerosis explained simply, without the hype and without the doom and gloom, just the science-backed truth, then you are absolutely in the right place. This is what we do here. Subscribe and hit the bell so you don't miss what's next. Thank you for your time and your trust. I'll see you in the next one.